Hi, good afternoon, everyone. We'd like to welcome you to our Summer Research Seminar Series here in HOPP. And we're excited to have you all join us. As a reminder, our format for today will have a couple of opening remarks. Then our speaker will share their lecture, and then we'll end with our Q&A session. To submit questions, you can enter them via the Q&A feature here on Zoom. And also another reminder, we will be sending surveys for attendees to complete. So we welcome you all to complete that. So our talk for today will be looking at bioinformatics. Our guest speaker is Dr. Christopher Fong. He's currently a bioinformatics software engineer for the MSK-MIND program, where he is assisting in research initiatives on precision medicine using multimodal data, integrating genomics, radiology, and pathology diagnostics. Before joining the MSK-MIND program, he worked under Dr. Niki Schultz as a postdoctoral fellow, analyzing large-scale genomic and clinical data for retrospective studies. Chris has a background in biomedical and electrical engineering that he's used towards various translational research projects, and he will highlight one of those projects today. Again, we'd like to welcome you all, and thank you so much, Chris, for speaking with us today.
Thanks for having me. So the talk is going to be on large-scale integration of genomic and clinical data. I know the audience has high school students. So, for this talk, I want to talk about cancer research, of course, but also, talk about getting insight on a career path, and just seeing what it's like as a bioinformatics software engineer. So I'll start off with the talking about, career-wise, what bioinformatics is. And so, this is a topic that combines, biology, mathematics, computer science, and medicine all together to help utilize what is known in the tools that are provided in computer science and modeling, mathematical modeling that you can do in physics with the mechanisms in biology and what has been done in medicine here. Sorry, my mouse is a little sensitive here. So, I kind of think of bioinformatics in two different realms, really, and especially at MSK, where you have a more genomics or molecular biology focused track here, and where bioinformatics is utilized here is to do the sequence calling. So you have a sample. And it goes through a sequencing machine, and there's an algorithmic process to actually determine what genes have been altered in a genomic sequence. And then you have like a very with the genomics involved, maybe you have a set of samples. You can determine the evolution or phylogeny of cancer or anything, of like COVID. And so, you can utilize bioinformatics in that way. And then there's also proteomics and how proteins and maybe RNA, transcriptome information that you can gather, how that, goes through that process. And so the career track that you would want, you can work for a pharmaceutical company like Genentech or Gilead, more commercialized or consumer-based companies like 23andMe or Ancestry, and of course, any research institution, hospitals and whatnot, like MSK. And then on the other side is more clinically focused informatics, So that has to deal with telehealth and healthcare applications. Just going through, there's a lot of documentation that's being generated by oncologists, radiologists, pathologists, et cetera. And so like digging through their doctor's notes, and we call this electronic medical records, EMR, where you can do data analysis using natural language processing to really dig into what happened in the patient's history. And then like the patient portals and data management, that's another aspect of the informatics. And so you can, with those skills, you can work for companies like Oscar Health, Flatiron. Epic is a huge system that's utilized at MSK. And then this right in the middle is like more translational bioinformatics, especially in oncology. I like to consider that like can look into clinical epidemiology, patient outcomes with this and precision medicine. So taking information not only about the medicine and how patient care has been done in the past, but how the mechanisms of molecular biology work in making like a specialized treatment plan for a patient. And then the other aspect is like a guidance system. So like, using NLP to find specific characteristics within text, or for radiologists, finding a tumor, having a guidance system, find the tumor within the MRI. And so, yeah, again, like research institutions for sure, like heavily are looking into this. They have a ton of data to work with. And then also pharmaceutical companies trying to figure out the next cure for cancer per se, or something like that. They need all this information here. And then healthcare systems. I mean, just like thinking about insurance as well, like what works, what doesn't work, what patients are being diagnosed. So just thinking about my own career path and how I got there. Again, like I studied electrical engineering, where I worked in the sensors lab, some more hardware-based, that led into working at a startup looking into medical sensors. And then a lot of, so much changed for me once I did my PhD. I learned about translational research, worked on imaging systems, and participated in and led in clinical studies really for these imaging systems. And any door that opened for me, I walked through. So I worked in strategy consulting a little bit for the tech transfer office, thinking about patenting and marketing for medical devices and whatnot, or anything dealing with healthcare. And also worked in more startups looking into signal processing and machine learning. And this is all prior to MSK. And if you have any questions on a career path, please let me know. Like, happy to give input or have my take on this. With all these skills that I acquired over the years, I started a postdoc and then worked in clinical data mining and cancer genomics. MSK has so much data to work with. The sky's the limit on what you want to research. So it's been a great opportunity. And I feel very fortunate, privileged to work here. And yeah, just to give a sense of like biomedical engineering, in case you want to major in that in college, there's multiple tracks: medical imaging, medical devices, that's sort of my realm, but also biomechanics and tissue engineering. And as a career path, a lot of people go into strategy consulting. Not so much from the medical imaging and medical device realm, but like if you've studied biomechanics and tissue engineering, it's easier to get into more less technical work than the technical aspects of these tracks here. Another, career path, equity research, so working more in finance or tech transfer, patenting offices and marketing and whatnot for medical or healthcare applications. And then finally, pre-med. A lot of the undergrads that I TA'd or mentored during my PhD were pre-med, like the large majority. So it is actually a pretty good major to look into if you're thinking about going into medical school down the line. And then so like for this talk, for the science part of this talk, it's more translational informatics. So really taking what is known from the mechanisms of molecular biology and cancer genomics and integrating that with the clinical aspects. That's really where you can answer important questions here, especially for clinical trials. So just digging a little bit deeper into that. These are some of the aspects again, and we'll focus on some of these here. What are the requirements for this? For translational bioinformatics, always start with an unmet clinical need. Sometimes people were just like digging into the data for sake of digging into the data, but a lot of what should be done is talking to having a collaborator, like a medical oncology fellow who knows and treats patients and is on the front lines and has a need that they need help with, that an engineer or a bioinformatics person can help with. And then you want to have a lot of comprehensive diagnostic data. What is great about MSK is that we have a lot of genomic data through MSK Impact, and I'll go in a little bit into that. And then there's a ton of, every patient has some clinical electronic medical health record or electronic health records. And of course, there's pathology and radiology images, but they're largely described in EHR. And so just focus on these two today. And of course, you need some quantitative skills that I talked about in the first slides. Okay, so again, the talk was large-scale integration of genomic and clinical data. So let's talk about a use case. And this is like, what genomic biomarkers exist for metastatic cancer? And I think others have gone through this before. I'll just go over quickly that cancer is a genetic disease, and the genomic breakup can be thought of as not just mutations, but there's all types of different alterations that occur in genomics. And so this is at the micro level, you can have like a deletion or an insertion or a substitution involved that could be like a mutation. But at the macro level, you can have whole deletions of the sequence just being truncated or duplicated here. And there's all types of different alterations that can occur, and it becomes very incredibly complicated. But these are all sorts of biomarkers for cancer here potentially. Just another basic slide on cancer itself. normal cell division has a normal cell cycle. Cells are created and apoptosis occurs, and so there's a normal cycle here of cells. But in malignant cell division, you may have some type of mutation or alterations that we talked about in the previous slide that causes abnormal cell cycle, and then the cells start to really just become just multiply uncontrollably. And the metastatic part, so you can have a primary cancer in the body, let's say in the colon, and that same cancer, those same set of cancer cells can move or spread to other parts of the body. And so liver and lung metastases are very common for patients with colorectal cancer. And this isn't like now like liver cancer or lung cancer. These are literally the same type of cells. So you have colon cells that end up in the liver and lung. So and these are the modes of transport that typically happen. Like cancer cells will travel through the blood or lymphatic system here. And then why is this important? So like some epidemiology here. So if you look at 5-year survival rate, so like patients after 5 years after diagnosis, what percentage of the patients will survive given their prognosis at the initial diagnosis? So if you have more localized cancer, so back to like the colorectal cancer example. So if you just have localized colorectal cancer, you have a 90% chance of survival after 5 years. Sorry. But if you have metastatic cancer at diagnosis, your chances of survival after 5 years is like less than 15%. And this is across all cancers. Like prostate cancer, if diagnosed somewhat or even early, or even it only has to have a little bit of spread, this is still 99%. So after 5 years, 99% chance of survival. Same thing with breast cancer here. It's like 98, 97%, I think. That's the number there. But it completely drops off when you have metastatic cancer. So you all have hopefully heard of retrospective studies. And so like having a large number of patients that have been treated for cancer, if just looking back at how that they were treated, what type of attributes that they had, their demographics or whatnot, what their stage at diagnosis are, all of that, you can begin to think about what biomarkers exist for certain cancers. And in order to do that, you have to process a large amount of data. And in genomics, it's pretty straightforward. A patient will have some initial surgery, more or less, to remove the cancer, or have a biopsy to look at the cancer itself. It goes through a genomic sequencing to process the sample. You have the sequence, and then you can quantify which alterations exist. And it becomes very structured. It's easy to tabulate all of this genomic data. But for clinical data, it's much more complicated. There are many different people that a patient will visit, will talk to at any given point in their cancer care. And so a lot of notes are being generated here. So like you have a lot of free text notes, and this could be saved to some database, but where things get really tricky is this manual chart review if you're doing a retrospective study, say you have 1,000 patients, you have to go through 1,000 patient charts, and each patient has tons of notes. So, this is a lengthy review process. But then at some point, specific info is being put into a spreadsheet. And yeah, so like when I first came here as a postdoc, I would meet these medical oncology fellows, and they would spend like nights, weekends, holidays, like it was just ridiculous for months just doing manual chart review. And so it was such a waste of time, and really, there's a lot of money that goes into cancer, and spending it on something so mundane is just ridiculous to me. So like I decided to go out and try to find more solutions on that. So and the hope is if we can use AI to remove the need for manual chart review, or at least accelerate it. And so, yeah, so this is just a summary. There's a lack of structure in clinical data. Comes from all kinds of different data sources. And then at the same time, you have this genomic cohort that is quickly growing. It's much easier to get genomic data than structured clinical data. Or there, at least at MSK, there's a very nice workflow. And then, so yeah, what are the key elements that we want to extract from this for our specific question? So metastatic events, the timing, what are the effective organ sites, and then separately, in a separate effort, we're looking into medication information. And then with this information, you can start to look into progression-free survival, how these patients, how a subset of patients fared given a different set of treatment. Okay, so the hypothesis really is that if we can do some large-scale integration of this clinical and genomic data, that we can properly find biomarkers for disease burden and metastatic risk. Okay, and I just want to give a quick shout out to the MSK team here. I definitely have missed some people. This was a really large effort. A lot of collaboration was involved. Really grateful to be on this project. Okay, so let me go through some of the methods for this project. Hopefully some of you know at this point about MSK Impact. It's a clinical sequencing platform looking into, so there's a targeted set of genes. Sorry, this needs to be updated here. There's more than 500 now at this point, and the latest assay. But it's a assay that looks at mutations, copy number changes in rearrangement, and sort of like what I showed prior. This is the workflow. The patient goes through a consenting process, surgery or biopsies happens, and have a match normal, which is blood, sequenced, and then eventually reviewed and signed out. And like, again, it's not trivial, but there's a great process at MSK where clinical reports are generated. You can put in these patients into clinical trials depending on what the genomic alterations that they have, and then all of this data goes into cBioportal, which I'll try to give a demo at the end of the talk here. So, okay, so when you go into cBioportal right now, and just thinking about like clinical data itself involved, there's not so much information, but you do know if a patient, you do know if a patient has the primary disease sequenced or the metastasis. So back to our colorectal example, the primary being colorectal cancer, so a tumor that's in the colon, a sequence metastasis, that would be like the tumor that for that colorectal patient that exists in the sample that exists in the liver or the lung. But so depending on where the patient is in terms of like in their stage of cancer, just knowing what the primary is or metastasis sample is, how it is genomically sequenced is doesn't give you much information. So so the sample status here can only take you so far. Like this is only really it's just a snapshot of what happens within the patient care. This is literally how the patient is at surgery, and not so much prior or within the future. So I always think of the analogy is that this is just a snapshot in time, but we really need the whole video. So we need to have the disease status for these patients at the moment of surgery. And so did the patient have metastatic disease? This isn't so known. I mean, like you may have surgery on a primary, but was the patient, did they have metastatic disease? You only know from this information that this 39% had metastatic disease. But so if you look into the clinical data, and let's say you do some like chart review from all of these different sources, you can get that information. Or you can go through a more quantitative approach and do data extraction. And so this is a process that we've started where we have this pipeline of structured data where we use natural language processing and ML models and to gather dates of procedure and metastatic events, sites of metastasis, and other information in pathology reports. And then we'll aggregate this data, providing a timeline of events, and then we can calculate metastatic free survival or get other demographic information and do the large scale analysis appropriate for retrospective studies. And just to drive this home, so like this is information really that if you go to CBio portal, just like the vanilla version, you'll get this amount of information, but really what you want is you want to know if a patient had a prior diagnosis, if they had leukemia as a child and then had breast cancer in their 70s or something like that. You want to know like they had a surgery or a biopsy, and when they had it relative to when they actually had the sample sequenced. Sometimes this can be years apart here. And then what metastatic events occurred on top of that. Okay. And then, so we have this pipeline, and we're creating structured data that I think would be more analogous to like an Excel sheet. But in order to visualize this data, we can transform it and put it into what's called CBio portal, if you're not familiar. And we can view all of this data. And so we're in a process of adding as much clinical data as possible, using the data mining techniques that have been come up with. Okay, so yeah, just back to real quick to like skills and career track. what's necessary in the clinical data mining portion. Well, I have to know programming, SQL, R. Those are really the backbone of doing this. You need to know some data science techniques. So data manipulation, so using pandas or something like that, which is library in Python, natural language processing, machine learning. And I would say biostatistics is like, if I could go back, I would definitely do a lot more or study a lot more statistics. I took like 7 semesters of calculus, partial differential equations, optimizations, but statistics ended up being like the most important out of all of those, especially for scientists. And from there, you can become a data scientist or data engineer. And talked about some companies before. More on the cancer genomics side, genomics, molecular biology, that's, the subjects of interest and then, the software involved. I honestly I'm not so much of an expert in these fields, but I would say that knowing biostatistics and data visualization is super helpful to being a good bioinformatics software engineer and computational biologist. Okay, then yeah, so some of the results. So like, we're trying to fuse a lot of different data, and so in order to get the patient history of metastatic events. And so these are the sources that we utilize in order to get that information. We know from the impact samples if it's a met or primary, and what the locations were for those samples. And then also through the data mining techniques, we know the data procedure. So we know when, at least through that, when they were had a metastatic diagnosis. So yeah, the pathology reports, then radiology reports doing mining, and then what's called ICD billing codes, so these are for insurance. And then the tumor registry, which is a database for cancer. And then real quick, this is like what the data really looks like within the tumor registry. So this patient had like a endocarcinoma of the lung, had started the stage 3, but then at some point had distant metastasis. This is what billing codes ICD billing codes look like. You have specific cancer diagnosis, but you have like metastatic cancer indicated here. And then radiology. This is all, free text. You can gather information about specific sites of metastasis through the findings section, and then really just gets summarized in the impression section. And you can find if there's indication of metastasis within the impression. And I want to just note that Anisha Luthra has been integral for this work here. And just real quick, just a couple of slides on just natural language processing in general. If you were to look at millions or at least hundreds of thousands of these reports, you can what is called vectorize these words into numbers. And so with some of the algorithms that are available, you can have a learned representation of the words within the documents, and represent them in say a 3D vector space, but it can be infinite dimensions. And so, but in a 3D dimension, if you take all the text in the world, let's say you scrape Google for all the information that's on the web, and do a learned representation, you can see that man is closely related to woman, the king is closely related to queen. Same thing with verbs, swam, swimming, or just locations in the world. And so, that's for any word in, like, if you were to take all of Google and figure out like what's closely represented, you can get like a scatter plot and see what's closely related here. But for cancer, it's a lot more tricky. And so if you want to do the same for metastasis, this is something that can be generated here. And so this is from a public data set that's available. And so if you look at what words are closely related to metastasis, this is what comes up. You can go one step further and look at what sentences are more closely related. There's a lot of sophisticated methods that I won't go into. But yeah, that's essentially NLP in a nutshell there. Okay, so let's think about the integration now. And I just want to point out that there's a paper in preprint. Yeah, so this is something that was submitted recently on this work, so I just wanted to plug that. Right, so then going back to converging disease status given like the clinical data mining pipeline. We have this here, but now if you've aggregated all of that clinical data and figured out, did the patient have metastatic cancer at the time of surgery for the impact sampling? This is what comes up. So you actually see that so like 12% of these patients don't have metastatic cancer, while the rest of them, of this 61%, at some point have metastatic cancer. So this is, again, this is no evidence of Mets in their clinical history. This is, these are patients that had metastatic cancer after their biopsy of the genomic sampling, and then the 33%, they had metastatic cancer at the time of surgery. And then, yeah, doing more data mining, we can see the number of patients with specific sites of disease here, and whatnot. I want to be able to get to the demo in a couple minutes, so I'm going to start going through this pretty quickly. So we can see the sites of disease, and then we can see the number of affected organs. So we could call this here metastatic burden. So, this project has been in flux, but this is just a cohort description. You can check the paper out to get the full details, but at the end of the day, we have more than 25,000 samples that were analyzed here. I'll just go through this quickly as a summary. This is a prior version of what's shown in the paper, but the results are still hold the same. This is really a resource to show what type of cancers are most prevalent at MSK. So, taken many different cancers and broken them into different specific disease types here with the sample counts. And then with that pie chart I showed earlier, we could see that which cancers are more prevalent to metastatic cancer, or conversely, that cancers that don't have as much evidence of metastatic disease, patients that fare a lot better. And then the sites of disease and its prevalence here. So so we have 19 organ sites here. I'll just go through and say that bone, liver, and lung are the most prevalent sites of metastasis in pretty much all cancers, but really across the board if you take an aggregate of all these 45 types. And then in breast cancer specifically, you can see, we can have break it down into three different subcategories here, and within those categories, you can see like the triple negative cases have less, are less prone to have bone metastasis compared to the other molecular subtypes. And similarly in colorectal cancer, cases where you have a high number of mutations, first of all, fare better than if you don't have a high number of mutations. But in the hypermutant cases, they're less susceptible to metastasize to the liver here. Okay, and then just real quick, some of the biomarkers. I want to talk about tumor mutational burden. I talked about hypermutants. So if you think about the number of mutations reported per megabase, this is what you get across all cancers. This is some work that was published in 2013. Basically, the point of this is to show that depending on cancer type, there's more prevalence of a high mutation. So melanoma has a much higher number of mutations than say like prostate cancer here. And then also the fraction of genome altered is the percentage of the genome that's been affected by copy number gains and losses. So earlier in the talk, I talked about duplications and deletions here. So this is a result of what happens there, and it's measured by FGA. So real quick, if you look at tumor mutational burden, you can see that in comparing metastatic samples versus primary samples, that do get a larger number of samples here a number of cancer types that have significant difference between metastatic and primary cases here. So, for example, lung cancer has many more samples with a higher tumor mutational burden or more mutations than their primary counterparts. But if you look at the fraction of genome altered, there's many more cancer types that where the metastatic sample has a much higher FGA than a significant more higher FGA than the primary counterparts here. And so with that, we say that copy number largely drives metastatic potential in not all cancers, but many cancers. So also if you look at the number of sites of disease, metastatic disease in patient history versus the FGA as well, you actually see a climb in the FGA. So like if a patient has patients that have no metastatic disease, have a median FGA of just above 0.2, but as you have more sites of metastatic disease, the FGA increases. And so, if you look at all samples here, all genomically sequenced samples, you can see an upward trend. But if you break down by the primary and metastasis, you see that the metastasis increases. But if you already have metastatic disease, the FGA doesn't really change so much here. So we thought that was an interesting case. It's still significant, but it's much flatter. So we thought that was interesting. And you can do this for all cancer types. really, and look at the correlation. So if you have a positive correlation, the trend goes up. If you have a negative correlation, the trend goes down. Negative correlation on this side, and so the cancer types that have a positive correlation and are significant are colored here. And yeah, so that's all I'll say on that. So yeah, all right, conclusions here. We have a clinical data pipeline to extract metastatic events and sites. Any attribute really that a physician would like, we can try to mine for that and put it from an unstructured format into a structured format. And with that, we've created this metastatic disease gradient, so like the heat map that can be used as a resource for impact samples. And then, through some of the research, we saw that higher chromosomal instability and metastases occurring compared to primary samples, but these differences aren't observed in all cancer types. And then also that there's a strong correlation between chromosomal instability and metastatic potential. Okay, so I talked about the science. I just want to leave real quick with some key things that I feel like have helped me with whatever success that I've had. And so I'll just go through these real quick. Is really, set short and long-term goals. If you don't do that, you won't know where you're going. And so it's really important to set goals here. Access. I feel like access is so much. I really, it was a privilege really to get a PhD, and I don't think a lot of people realize that. Not many people have the opportunity to get that far, and I'm so grateful to have that. And it's really opened some doors, and I hope to open doors for others down the road. So access is really important. In order to get access, at a young age, I encourage everyone to network. So join clubs, sports, organizations. It's really important to know what, to get to know people and to know what they're doing, and make those connections and really connect with people. And I would say that staying curious leads to innovation. So never stop asking questions. When you stop asking questions, you really plateau. And in any project, so stay curious. I'd say be a leader, or know what it takes to be one. If you have a good idea, if you have any idea, and you want things to really blossom around that idea, be your own cheerleader and lead the way. And if you're not comfortable being a leader, at least know what it takes to be one, because you'll never know when you'll have to step up. A lot of my early mentors were athletes, so I learned too late that how hard they really had to work and persevere to get where they are. You think about exceptional performers, LeBron James, Kobe Bryant. They all talk about how hard work really got them to where they be. They have ridiculous schedules every single day, very regimented. So if you do what you love, make sure you work hard and just keep going. And there's always going to be pushback, or just we'll fall along the way, but keep going and persevere. And the most important thing in my mind is to stay in the moment and to practice mindfulness. There's nothing worse than to be taking your SATs or something like that and be thinking, I hope I get into the school I want, or I should have studied harder. Yeah, it really does nothing for you, and being in the moment can get you through the task at hand. So just want to point that out. I was going to do a demo, but do we have time for that, or should we jump to questions?
Sure, you can do the demo. It's fine. We don't have that many questions.
Okay. Okay, great. So I've already put this up here. I have a link here. And so everyone can get onto cbioportal.mskcc.org. You can view all of the genomic samples sequenced in MSK-Impact, like right now. And there's a ton of other studies that have been, these are like more or less public studies, or published studies rather, that have been put out by MSK. I'll show a private instance of MSK-MET. And so, unfortunately, you don't have access to this right now, but hopefully soon in the future. And I was going to go through what you can do here. And really, so this is all the data from the paper. I mentioned more than 25,000 patients, and we have all these different cancer types here. And then you can look at the other clinical information. So if the patient had a distant liver met, or what organ site system was affected, or if the patient had a history of metastatic disease in the clinical realm, but also like the genomic data as well, of course. So like mutated genes and the copy number changes involved. And we talked about FGA versus TMB. This is a scatter plot here. So if you want to compare like metastatic patients versus non-metastatic patients, we talked about the epidemiology before. So if you would click compare, I already have something up here. You can go to the survival tab, and you can see the percent survival for patients that had no metastatic disease history versus ones that did. So you can see the median survival here doesn't even hit 50%. But half of the patients with metastatic disease had it's actually down here. The median is 33 months here. So that's like one comparison that you can do. If you want to look into just the metastatic patients, and then to look into compare liver metastases, you can see, and then click to like genomic alteration. So metastatic patients, patients that did not have a liver met versus if you did have a liver met, and you look at the significant differences here for these different genes. Well, sorry, here you can see ESR1 is one gene that's reported to be significant. So patients that have ESR mutations tend to have a higher occurrence of liver mets, and that's actually something that was reported in the paper here in figure 4. And then finally, examples. So if you were to look into different race categories here, white, Asian, Black, or African American, and it takes a while to load. But actually, so if you look into breast cancer itself, so these are breast cancer patients comparing sorry, that's not the right figure here. So comparing Asian, Black, or African American or white, this is like something in the realm of disparity research that you can see that actually Black and African American has a significantly less survival than white and Asians. We have genomic information, of course, to look at these differences, but there's a lot of moving parts involved. So like insurance or occupation or just, income status and whatnot. So we don't have that data, but I hope to see that in the future. So I just wanted to point that out, that this kind of information can be looked at in CBio portal. And that's it. So that's all I have. Swanning knowledge, collaborators, especially Bastion, was very, really carried the way for this project. And of course, people in the MSK-MIND program. So thank you.
Great, thank you so much, Chris. We do have a couple of minutes left, so I'll share some questions with you. So the first is Murat. They ask, you work with a large amount of data that you have to analyze manually. Can you implement similar searching algorithms like Google, etc.?
Okay. Can you implement similar search algorithms like, yeah, so that's essentially what natural language processing is like. So so Google is really big with natural language processing, and that's actually, natural language processing is the backbone of, like, when you start typing something into Google and it auto-populates, that is like a natural language processing system involved in the back end. So we're essentially implementing similar Google-like algorithms for cancer.
Okay, great. Thanks so much. The next question was from Dominique. They asked, when you gather patients, doctors' notes in your clinical analysis, are there ever instances when some of the notes have conflicting ideas regarding diagnosis and treatment plans? And if so, how do you deal with that?
Yeah, so we don't really, so that's a good question. We know that conflicts exist. And some patients have incomplete information altogether. And so it's difficult to take into account mathematically, but statistically, that's something that you'll have to put into your algorithms. But what really solves that problem is a lot of data. So yeah, that's where really big data occurs. Not every patient, and rarely patients will have such conflicting diagnoses. Those, I think, are outlier cases. But when you have big data, you get to see the trends. And so when you're thinking about finding biomarkers for cancer, you're really like looking at what the majority, what the result regresses to, right? When you think about like finance, these same machine learning processes are applied in finance, but companies don't have the 100% perfect accounting. But these people in finance are finding ways to figure out what stocks to purchase. So like that they're doing something right, and they just happen to have a lot of data to back up their algorithms.
Okay, great. Thank you so much, Chris, for today's talk. We greatly appreciate it. And as a reminder for all of our attendees, we have a final lecture next week, Wednesday, at 1 p.m. And we look forward to having you all join us. Thank you again, Chris. You have a good rest of the day, everyone.
Thank you.